We're talking about carbohydrates, and I know as soon as I say that word, you're probably thinking about bread, pasta, the keto diet, or maybe that post-lunch slump.
It's inevitable, isn't it? The word carbohydrate has been almost entirely co-opted by the diet industry. It's become a buzzword for, calories we should avoid or fuel we need to manage.
Exactly. But today, we are tossing the diet books out the window. We aren't here to talk about waistlines.
Not at all.
We are here to talk about the molecular machinery of life. We're doing a comprehensive deep dive into Chapter 15 of Carbohydrates of Physiological Significance by Bender and Mays. And the goal is to look at the source code.
We're going to strip it right down to the atoms. Because when you stop looking at carbohydrates as just food and start looking at them as biochemistry, you realize they are, well, arguably the most versatile molecules in the human body.
And to set the stage, the text introduces a term right at the beginning that I think frames this perfectly. We all know genomics, the study of genes.
Right.
We know proteomics, the study of proteins. But this text opens with glycomics.
It's a term that really signifies a shift in how we understand biology. The glycome is the entire complement of sugars in an organism, and it is massive. It's not just the glucose floating in your veins.
So it's more than just fuel.
Oh, much more. It's the free sugars, the complex chains, the structures attached to proteins and lipids. It's everything.
The text actually defines it as the comprehensive study of the genetic, physiological, and pathological aspects of carbohydrates. That suggests that sugars are doing a lot more than just burning in a furnace.
Precisely. That is the core theme of this deep dive. Carbohydrates are information carriers. They are a biological language.
A language?
Yes. Depending on their shape, how they link together, and even which direction a single atom is pointing, they encode biological data.
So we aren't just talking about fuel tanks. We're talking about a communication network. But before we get into that complex language, we have to establish the basics. The text starts with a very bold, very simple claim.
It does.
Glucose is the most important carbohydrate.
It is the absolute king. In the metabolic hierarchy of mammals, glucose is the gold standard. It's the major metabolic fuel. It's the universal fuel for the fetus. There's no escaping it.
And the text traces this back to the very beginning of the energy cycle photosynthesis. Plants take CO2 and water, use sunlight, and synthesize glucose.
And we eat the plants, or we eat the animals that ate the plants. It's the foundation.
And here is the crucial physiological point, right?
Yeah.
No matter what form of carbohydrate you consume, whether it's the starch in a potato, the sucrose in a candy bar, or the lactose in a glass of milk.
The body's ultimate goal is almost always to convert it into glucose.
It's the universal currency.
It is. The liver acts as the central bank. It takes all these different incoming currencies, fructose, galactose, and so on, and it exchanges them for glucose to be released into the blood.
And when that machinery breaks, when that conversion process fails, that is when we see significant disease.
Absolutely.
The text specifically lists diabetes mellitus, which everyone knows, but it also lists galactosemia, glycogen storage diseases, and lactose intolerance.
Yeah.
These aren't just, stomach aches.
No, these are fundamental failures in the molecular machinery we are about to discuss.
Exactly. So to understand why the machine breaks, we have to understand how it's built. We need to look at the blueprints.
Let's do it.
Let's jump into Section 1, the classification of carbohydrates. The text breaks them down into 4 main groups, and I want to go through these carefully because the terminology really matters here.
It does. We start small and get big. The first group is monosaccharides. Mono meaning one.
The building blocks.
The fundamental units. Biologically, these are sugars that cannot be hydrolyzed or, broken down by water into simpler carbohydrates.
So these are the individual Lego bricks.
Correct. If you smash a monosaccharide, you don't get sugar anymore. You just get carbon, hydrogen, and oxygen atoms. It's the smallest possible unit of sugar.
Okay, then we have disaccharides.
2 monosaccharides snap together. Di for 2. These are condensation products. Examples would be lactose or sucrose.
Got it. Then oligosaccharides.
Oligo is Greek for a few. Usually this refers to chains of 3 to 10 monosaccharide units.
Okay, a small chain.
Right. And the text makes an interesting note here. Most of these are not digested by human enzymes.
So they pass right through us.
Often, yes. Or they become food for our gut bacteria rather than fuel for us. They're still important, just not in the way we usually think of carbs.
And finally, polysaccharides.
Heavyweights, poly for many. Condensation products of more than 10 units. This is your starch, your glycogen. These can be massive, containing thousands of units, and they can be linear chains or highly branched structures.
Okay, I want to double-click on the monosaccharides. The text dives into some pretty dense chemistry here regarding aldoses and ketoses. It defines carbohydrates as aldehyde or ketone derivatives of polyhydric alcohols.
Right.
Now, for a listener who hasn't been in a chemistry lab since high school, can we visualize what that means?
Absolutely. Let's build a mental model. Imagine a backbone of carbon atoms, just a vertical line of carbons holding hands.
Got the line, a carbon chain.
Attached to these carbons are hydrogen atoms and hydroxyl groups. That's an oxygen and a hydrogen, or 8OH. That's the polyhydric alcohol part, lots of hydroxyls.
Okay.
But the defining feature of any sugar, the thing that makes it a sugar, is a specific cluster called a carbonyl group. That is a carbon atom double bonded to an oxygen atom.
Okay, carbon double bonded to oxygen. That's the landmark.
The position of that landmark determines the entire personality of the sugar. If that carbonyl group is at the very end of the carbon chain, like the head of a snake, it is an aldehyde group. So we call that sugar an aldose.
End of the chain equals aldose. Simple enough.
Exactly. Now, if that carbonyl group is tucked inside the chain, attached to a carbon somewhere in the middle, it is a ketone group. So we call that sugar a ketose.
And this isn't just a naming convention. This actually matters.
Oh, it changes the chemistry completely. It's a huge deal. Aldehydes and ketones react differently with other molecules. The text refers us to Table 15.4, which lays this out beautifully. Let's look at the trioses, the 3 carbon sugars.
Okay, looking at the table, we have glycerose, also called glyceraldehyde.
That is the aldose. See how the double bond is at the top on carbon 1? At the very end.
Yep, I see it. And then next to it is dihydroxyacetone.
That is the ketose. The double bond is on carbon 2, right in the middle of that 3-carbon chain.
And chemically, they have the exact same formula. C3H6O3, same number of carbons, hydrogens, and oxygens.
Identical formula. But because that double bond moved one step down...
It's a completely different molecule.
Completely. And this pattern repeats as you add more carbons. Look at the pentose is 5 carbons. Ribose is your aldose. Ribulose is your ketose.
And the big ones. The hexoses, 6 carbons.
This is the most famous pair. Glucose is an aldose. Fructose is a ketose.
That is a huge light bulb moment. I've always known glucose and fructose were different, but I didn't realize fructose is essentially just the ketone version of the 6-carbon sugar.
That's exactly what it is. And that structural difference, having the reactive group in the middle rather than the end, is why your body processes them so differently, why they taste different, and why they have different metabolic effects. It all comes back to that one detail.
Okay, keeping with this theme of structure is destiny, we have to talk about the shape of glucose. Section 2 of our outline focuses on figure 15:1. The text presents glucose in 3 completely different ways. It's almost like a police lineup of the same suspect in different disguises.
It's a critical concept because molecules are 3D objects, but we are stuck drawing them on 2D paper. Figure 15:1 starts with the A, straight chain structural formula.
This looks like the ladder we just described. Vertical carbon backbone.
Right. This is the aldohexose form. You can see the aldehyde group at the top. But here's the reality check. Glucose is kind of claustrophobic. In a solution, like in your blood, it acts to stabilize itself. It doesn't like to stay in that open straight line.
So what does it do? It can't just stay like that?
It folds. It bites its own tail. The aldehyde group at carbon 1 reaches down and reacts with the hydroxyl group at carbon 5. Forming a ring. This is called a hemiacetal formation. And the text notes that this cyclic structure is thermodynamically favored. In fact, 99% of the glucose in a solution is in this ring form. The straight chain is actually quite rare.
Which leads us to the second drawing in the figure, the Peehe-Worth projection.
This is the classic hexagon you see in every biology textbook. It represents that cyclic ring. It's drawn flat as if you were looking at a dinner plate on a table from an angle.
And the bonds nearest to you are drawn with thick, bold lines.
Right, that's to show perspective, to give it a sense of 3 dimensions, even though it's flat.
So that shows us the connections. But the text says even this isn't the whole truth.
It's not. Because of physics, carbon atoms have very specific bond angles, about 109.5 degrees. If you try to force 6 carbons into a perfectly flat hexagon, you create a lot of strain on the bonds. They want to snap.
So they buckle to get comfortable.
They buckle. And that is drawing C, the chair form. Imagine a beach chair. You have a backrest tilting up, a seat in the middle, and a footrest tilting down.
I can picture that zigzag shape.
That is how the glucose ring actually sits in 3D space. It puckers into that chair shape to relieve the strain. It is the most stable, lowest energy conformation.
It's amazing how much detail goes into just existing. But the text introduces another layer of complexity here that we absolutely have to cover. Isomerism. specifically D and L forms.
This is where we get into handedness, the concept of chirality.
The text mentions figure 15-2, contrasting D-glyceraldehyde and L-glyceraldehyde.
Think of your hands. They have the same components. Palm, 4 fingers, 1 thumb, but they are mirror images. You cannot superimpose them.
Right, you can't put a left-handed glove on a right hand.
Exactly. And sugar molecules work the same way. The D and L designation is based on the spatial relationship to the parent compound, glyceraldehyde. You look at the carbon atom adjacent to the terminal alcohol group. In glucose, that's carbon 5.
Okay, looking at carbon 5.
If the hydroxyl group on that carbon is pointing to the right side in the flat projection, it is the D isomer. If it is pointing to the left side, it is the L isomer.
And does this matter to my body, or is this just something chemists argue about?
Oh, it matters more than almost anything. Most naturally occurring monosaccharides are D sugars. Your body's enzymes, the machines that process sugar, are built with this specific geometry. They are right-handed gloves, so to speak.
So if I ate L-glucose...
Your body wouldn't recognize it. It wouldn't fit into the enzymes. It would just pass right through you. This is metabolic specificity at its most fundamental. We live in a D-sugar world.
That is wild. Now, the text warns about a potential point of confusion here regarding optical activity in Section 3.
Right. This is often where students get tripped up, and it's a good point to clarify. Because these carbon atoms are asymmetric attached to 4 different things, they interact with light in a weird way.
How so?
If you shine a beam of polarized light through a sugar solution, the sugar will physically rotate the beam of light.
Like twisting a dial.
Exactly. If it rotates the light to the right, it is dextrorotatory, and we use a plus sign, plus.
Yeah.
If it rotates it to the left, it is lavorotatory with a minus sign.
But the confusion is?
But the confusion is that D and L refer to the structure, the physical shape, the handedness. The plus and on blanche refer to the optical rotation, which is a physical property. They usually match up. D sugars usually rotate right, but not always. You can absolutely have a D-shaped sugar that rotates light to the left.
Got it. So DL is the architecture, plus MS is the behavior.
Perfectly said. That's a great way to put it.
Now let's go back to those rings. The text mentions pyranose versus furanose rings in figures 15-3 and 15-4.
This is simply about the size of the ring that forms. If the sugar forms a 6-membered ring, which is 5 carbons and 1 oxygen atom, it chemically resembles a substance called pyrin, so we call it a pyranose.
And furanose must be a 5-membered ring.
Exactly. 4 carbons and 1 oxygen atom, resembling a molecule called furan.
And does glucose have a preference?
Glucose is almost always a pyranose. We call it glucopyranose. But fructose, interestingly, often forms a furanose ring, a fructofuranose. That smaller 5-sided ring is characteristic of fructose in many important biological compounds like sucrose.
Okay, now we arrive at what I think is the most technical but perhaps most structurally important concept in the chapter, alpha and beta anomers. The text points to figure 15-4. What are we looking at here?
We are looking at the anomeric carbon. Remember when the straight chain bit its own tail to form the ring?
Yes, the reaction between carbon 1 and carbon 5.
When that ring closes at carbon 1, a new hydroxyl group, OH, is formed at that junction. And that group has a choice. It can get locked into position pointing down relative to the plane of the ring, or it can get locked into position pointing up.
So it's a coin flip during the reaction.
Basically, in the alpha form, the hydroxyl group on that anomeric carbon is on the same side as the ring oxygen. Conventionally, we draw it pointing downwards. In the beta form, it points upwards.
I think I found a mnemonic for this once.
Oh.
Alpha falls, beta balloons, alpha points down, beta points up.
That works. That's a great way to remember it.
Yeah.
And in a solution, these forms are constantly flipping back and forth. This is called mutarotation. For glucose, you eventually settle at an equilibrium of about 38% alpha and 62% beta.
Why do we care about this tiny flip? It seems so minor.
We care because biology cares. That tiny difference, whether that bond points down or up, is going to be the difference between a potato and a tree trunk later in this deep dive. It dictates everything about how sugars link together.
Okay, foreshadowing. One last shape-shifting concept before we move to the specific sugars. Epimers, figure 15-5.
Epimers are isomers that differ at just one specific carbon center. Basically, if you take glucose and you flip the EpOH group at just carbon 2 from one side to the other, you don't have glucose anymore. You have a new sugar called mannose.
And if you leave carbon 2 alone but flip the one at carbon 4,
You get galactose.
so biologically, mannose and galactose are just Glucose with a twist.
Structurally, yes, they are the most important epimers of glucose, but physiologically, that one little twist makes them distinct keys for distinct locks in our metabolic pathways.
All right, we've built the vocabulary. We understand aldose versus ketose, rings, anomers, and epimers. Let's look at the roster of players. Section 4, important monosaccharides.
Let's start with the pentoses, the 5-carbon sugars in table 15-2. The absolute star here is D-ribose.
Ribonucleic acid, RNA.
Exactly. Ribose is the R in RNA. It is the structural backbone of our genetic machinery. It's also the backbone of ATP, the energy currency of the cell, and NAD, a crucial coenzyme. You literally cannot have life as we know it without ribose.
The table also mentions L-xylulose. That's a strange one.
This is a niche one, but interesting. There's a benign genetic condition called essential pentasuria. People with this condition excrete L-xylulose in their urine because they lack the specific enzyme to process it.
So it's harmless.
Generally harmless, but it illustrates just how incredibly specific our metabolic pathways are. There's one enzyme for this one sugar, and if it's missing, the sugar has nowhere to go.
Moving to the hexoses in table 15-3, these are the heavy hitters. We've talked about D-glucose blood sugar.
Right, and the clinical note here is glucosuria. In poorly controlled diabetes mellitus, the blood glucose level gets so high that the kidneys can't reabsorb it all and it spills into the urine.
Then we have D-fructose.
Found in fruit and honey. It is famously sweeter than sucrose, but there is a serious condition mentioned: hereditary fructose intolerance.
What happens there?
If you lack the enzyme to break down fructose, unprocessed fructose phosphates accumulate in the liver and kidneys. This actually causes a severe drop in blood sugar hypoglycemia.
That sounds completely counterintuitive. You eat sugar in the form of fructose, but your blood sugar drops.
It happens because the accumulated fructose metabolites inhibit the process of breaking down glycogen. They essentially create a metabolic traffic jam that stops the liver from releasing its stored glucose into the blood. It can be very dangerous, especially in infants.
Wow. And D-galactose.
This is a component of milk sugar derived from the hydrolysis of lactose. It's also synthesized in the mammary glands to make milk for newborns.
And the disease connection, galactosemia.
Hereditary galactosemia. This is a failure to metabolize galactose. The galactose builds up in the blood and tissues. The text notes specifically that it can cause cataracts.
Cataracts in babies, how does that work?
It's the direct chemical consequence. The excess galactose is converted by an enzyme in the lens of the eye into an alcohol form called galactitol. Galactitol can't get out of the lens, so it accumulates, draws in water by osmosis, and clouds the lens. It's a perfect example of how a molecular defect leads to a visible physical symptom.
That's incredible. Let's move to section 5, derivatives. We aren't just talking about plain sugars anymore. We are talking about modified sugars.
Nature is a great chemist. It loves to swap out parts. One major class mentioned is glycosides. This happens when the hydroxyl group on the anomeric carbon, that alpha-beta spot, condenses with another compound.
And the non-sugar part is called the aglycone.
Right, and these have huge medical relevance. The text discusses cardiac glycosides. These are derivatives of plants like digitalis or strophanthus. Ouabain is the example given.
These are heart medications, right?
Powerful ones. They inhibit the sodium-potassium pump in the cell membranes of heart muscle. In the right dose, they increase the force of heart contraction, which is great for heart failure. In the wrong dose, they are deadly poisons.
And streptomycin?
An antibiotic. It's a glycoside, too. So these structures are all over pharmacology.
Then we have deoxysugars.
Deoxy literally means missing an oxygen. Figure 15-9 shows 2 deoxyribofuranose,
Which is the D in DNA. Deoxyribonucleic acid.
Precisely. It is ribose, but the hydroxyl group at carbon 2 is replaced by a simple hydrogen atom. That one missing oxygen atom makes DNA chemically much more stable than RNA. It's the reason our genetic history can be preserved for thousands, even millions of years. That one atom.
And amino sugars.
Here you replace a hydroxyl group with an amino group. Addition NH2. Figure 1510 shows glucosamine. You've probably seen this in bottles at the pharmacy for joint health.
Right, for cartilage.
Exactly. It's a major constituent of things like hyaluronic acid and other glycosaminoglycans that cushion our joints.
The text also mentions erythromycin.
Another major antibiotic. It contains amino sugars. So again, carbohydrates aren't just fuel. They're structural components and potent bioactive molecules.
Okay, let's scale up. Section 6, disaccharides. 2 sugars linked together.
The bond is called the oglycosidic bond. Figure 1511 breaks down the big 3 we encounter in our diet.
First up, maltose.
Glucose plus glucose. This is linked by an alpha-1 to 4 bond. This is what you get when your digestive enzymes like amylase start breaking down starch.
Second, sucrose, table sugar.
Glucose plus fructose. Now, the text includes a fascinating chemical nugget here. Sucrose is called invert sugar.
Why? What does it invert?
It goes back to that optical rotation we talked about. Sucrose itself is dextrorotatory. It rotates polarized light to the right, plus, plus. But if you hydrolyze it, split it into glucose and fructose, something interesting happens. Glucose rotates right, plus, but fructose rotates strongly to the left.
The fructose overpowers the glucose.
It does. The final mixture ends up rotating light to the left, so the rotation inverts from right to left. That's why honey, which is a mix of free glucose and fructose, is levorotatory.
There's a very cool detail, and practically the text mentions sucrase deficiency.
Same principle as the others. If you lack the enzyme sucrase, you can't split the bond. The sucrose sits in your gut, draws water in, which causes osmotic diarrhea, and then gets fermented by bacteria, which causes flatulence.
Which brings us to the third one, lactose.
Glucose plus galactose. But look at the bond in figure 1511. It's a beta 1 to 4 linkage.
A beta linkage, not an alpha.
This is the key. You need a specific enzyme, lactase, to cut a beta 1 to 4 bond. Most mammals stop producing lactase after they're weaned. Humans are unique in that some populations evolved to keep producing it into adulthood, the so-called lactase persistence trait.
And if you don't have that trait,
You have lactose intolerance. It's not an allergy. It's a simple enzyme deficiency.
it's amazing that a single bond orientation alpha versus beta dictates so much of our global diet.
There is one more disaccharide mentioned. Trehalose. Glucose plus glucose, but linked alpha-1,1. It's found in yeasts and fungi, but interestingly, it's the main sugar in the hemolymph of insects.
Insect blood sugar, I'll remember that. Now let's go big. Section 7, polysaccharides. This is about storage and structure.
The two main storage forms of glucose are starch, in plants, and glycogen, in animals. Both are homopolymers of glucose, just long, long chains of glucose.
But they're structured differently. Let's talk about starch first.
Starch has two components, amylose and amylopectin. Amylase, which is about 13–20% of starch, is a non-branching helical structure. Just a long coil of glucose units linked by those alpha-1,4 bonds we saw in maltose.
And amylopectin.
Amylopectin is branched. It has the same alpha-1 to 4 backbone, but every 24 to 30 residues, there is an alpha-1 to 6 bond that starts a new branch off the main chain.
Figure 1512 shows this branching clearly. Now compare that to glycogen, the animal starch.
Glycogen is like amylopectin on steroids. It is highly, highly branched. The chains are shorter, only 12–15 residues long, so the branches happen much more frequently. It's a much more compact, bushy molecule.
Why the difference? Why do animals need more branches than plants?
Think about the lifestyle. A plant, for the most part, sits in the sun. It has a steady, slow income of energy. It doesn't need to run away from anything.
But animals do.
Animals have fight or flight. When a predator jumps out, your muscles need a massive amount of glucose immediately.
And the enzymes that release the glucose can only work from the ends of the chains.
Exactly. Enzymes can only release glucose from the non-reducing ends of the branches. If you have a long, straight chain like amylose, you have only two ends to work from. It's a slow release. But if you have a highly branched bush like glycogen, you have thousands of ends. You can mobilize massive amounts of fuel almost instantly.
So the molecular architecture is dictated by the evolutionary need for speed. That is fascinating.
And looking at figure 1512, do you see the small dot in the center of the glycogen molecule, labeled as a primer?
Yes, the primer molecule.
That is a protein called glycogenin. You can't just start building a sugar chain from thin air. You need a foundation. Glycogenin acts as the seed. The glucose chain grows outward from it, forming these spherical granules called beta particles in your liver and muscle cells.
There is one other plant polysaccharide mentioned, inulin.
Not to be confused with insulin, inulin. It's a polymer of fructose found in tubers and dahlias. It is highly soluble, but human enzymes cannot hydrolyze it. It's a fiber.
So why is it useful medically?
Because it passes right through the kidneys without being reabsorbed or secreted. Doctors can use it to measure the glomerular filtration rate, or GFR. You inject a known amount and then measure how fast it's cleared into the urine. It's the perfect inert marker for kidney function.
Now, we arrive at the structural polysaccharides, figure 1513, and the big one, cellulose.
The most abundant organic substance on Earth, the chief constituent of plant cell walls, wood, cotton, paper, it's all cellulose.
Structurally, it is just glucose, same as starch.
Chemically, yes. The monomole is glucose. But structurally, it is completely different. Starch uses alpha 1 to 4 bonds. Cellulose uses beta 1 to bonds.
That beta flip again.
That one difference changes everything. Because of the beta orientation, the glucose chains in cellulose are straight and rigid. They aren't coiled. They can stack on top of each other, bonded by hydrogen bonds, forming these incredibly strong microfibrils. It's like rebar.
And digestion.
Mammals lack the enzyme to hydrolyze the beta 1 to 4 bond. We have amylase for the alpha bonds in starch, but we have absolutely nothing for the beta bonds in cellulose.
So we eat a potato, which is starch, and get energy. We eat wood, which is cellulose, and get nothing.
Just fiber. Bulk. Herbivores like cows and termites can only eat grass and wood because they harbor microorganisms in their gut that do have the enzyme cellulase to break that bond.
It really highlights the precision of biology. A tiny geometric shift transforms a primary fuel source into one of the strongest building materials in nature.
It defines entire food chains. It's why we can't live on grass.
What about chitin?
Chitin is the structural polysaccharide of the exoskeleton of crustaceans and insects. It's also found in the cell walls of fungi. It consists of N-acetyl-D-glucosamine units. It's incredibly tough, another great building material.
And pectin. That's what makes jam sack.
Exactly. It's found in fruit. It's a polymer of galacturonic acid, which is a modified form of galactose. It forms a gel structure, which is why we use it as a gelling agent.
Finally, let's discuss Section 8, complex carbohydrates. We are moving beyond just sugar chains to molecules that are hybrids.
This returns us to the glycome concept we started with. We have glycosaminoglycans, or GRANGE. These are complex carbohydrates containing amino sugars and uronic acids. They're often sulfated, which gives them a strong negative charge.
And they attach to proteins to form proteoglycans.
Think of this as the ground substance, or the stuffing of your connective tissues. Figure 1514 shows hyaluronic acid, chondroitin sulfate, and heparin.
Hyaluronic acid, that's in every skincare ad these days.
Because it binds massive amounts of water. It's a super absorbent polymer. In the body, it acts as a lubricant in synovial fluid in your joints and as a shock absorber in cartilage. It keeps tissues hydrated and plump.
And heparin?
An anticoagulant. It prevents blood clots. It is a highly sulfated JAG. It's a sugar derivative that saves lives in hospitals every single day.
Then we have glycoproteins on cell membranes. The text mentions sialic acids.
These are the flags on the outside of the cell. Figure 1515 shows N-acetylneuramic acid, which is the most common sialic acid. These sugars sit at the very ends of oligosaccharide chains on the cell surface.
This creates the glycocalyx.
The sugar coat. About 5% of the weight of a cell membrane is carbohydrate, and it's all on the outside, facing the world. It's the cell's identity. It allows cells to recognize each other.
The text mentions lectins here. What are they?
Lectins are proteins that bind to these specific sugar residues. It's a recognition system. It's how your immune system recognizes a pathogen, or how a sperm recognizes an egg, or how cells adhere to one another to form tissues.
And glycophorin.
It's a major integral membrane glycoprotein of human red blood cells. It spans the lipid membrane with its carbohydrate chains dangling outside. These chains are what carry the A, B, and O blood group antigens. Your blood type is determined by the sugars on your red blood cells.
So if we synthesize all of this, we started with a simple carbon chain. We curved it into a ring. We flipped an OH group up or down to make it alpha or beta. We linked them into chains to make starch for energy or cellulose for structure. And by the end, we have a system that not only fuels the furnace, but acts as the structural steel of the body and the ID card for every single cell.
That is the beauty of biochemistry. It's modular. You take a simple unit glucose, and by tweaking the geometry, the handedness, and the linkage, you get an almost infinite variety of biological tools.
It really is a code. The text says it explicitly.
Yeah.
The conformational variations encode biological information.
Just like the A, T, C, and G of DNA encode genetic information, the glycome, with its different sugars and linkages, encodes functional and structural information. It's a second language of biology.
Well, I have to say, my perspective has shifted. I won't just look at a potato as a carb anymore. It's a bundle of alpha-linked glucopyranose units waiting to be mobilized by my amylase.
And don't forget the chirality. It's all D-glucose.
Never forget the chirality. Thanks for guiding us through the molecular architecture of the glycome.
My pleasure. It's a beautiful subject.
And to you listening, next time you look at a nutrition label, look past the total carbs number. Think about the incredible molecular machinery spinning inside you to make sense of it all. Think about the handedness of the sugar in your food. See you in the next deep dive.